Right. Good morning, everyone. So, what we did yesterday was we just prepared a whole set of proteins, and I'm going to do a bit more of that in the second session today. So, I've got some outstanding proteins that need to be dealt with. And of course, yesterday we did a first docking experiment, although we haven't seen what the outcome is of that docking experiment. Okay. So the way we work is that once you've got the protein in, for now, because we don't know much about the active site and waters in the active site, for now we're just blindly deleting all waters just to make sure there's plenty of room within the protein for us to place our own ligand within that. And of course, we've removed some non-standard molecules that are not going to interfere with our, that could possibly interfere with our molecular docking, so we've removed them. Excuse me. And so we've removed DMSO, and I suspect that DMSO is an artifact of a crystallization process, so we have got that through. And then what we did is we saved the co-crystallized ligand to a place because it's a reference point. It is beautifully docked into the normally the active site of the protein. It's beautifully docked there. If we want to do molecular docking for maybe redock the ligand, and we need that point of reference. So we need it available to compare. And if we're redocking, we are validating the docking procedure that we are performing. Okay. Is there a question, comment? Okay. And once we've saved the co-crystallized ligand, we remove it, and then we save what's left. The apoprotein is left. We save that. We can save it as a PDB, for instance. But just over here, the co-crystallized ligand, when you save it, you can look, if you open it in Notepad++, you can see X, Y, and Z coordinates, and that gives you a really good idea of where the active site is for molecular docking. Okay. Please, we had a strange situation. I really wanted to give you a problem protein because you don't always have to do this. Not all the proteins you get from RCSB have more than one conformation. But if you try and work on the raw protein from RCSB, you may run into trouble. And of course, what we did to sort that trouble out is we just went to the cluster, and we were able to use MGL tools on the cluster to prepare PDB split old conts to split it into two conformations. So what you could do, this is a slightly different way of doing it. In the notes, what I've done is I've taken the protein and I've deleted everything, then saved it, taken it to the cluster, and then split conformations. What I've asked you to do is just split the conformations right at the start, and then bring back one of them and delete what we don't need. So there are two ways of doing things. But please, if you have a look at the proteins from RCSB, this is the kind of situation you see that alerts you that there are more than one conformation in it. So you can see these atom number 570 to atom 573. Okay.
Prof?
Yes?
I think there's something wrong with your microphone.
Okay.
I don't know if
is that better? Is that better? Hello? Can you hear me?
Yes, I can, but
is it?
I think it's got a sound that's disturbing.
That's is everyone else? All the setup is fine here. Is it like a noise sound?
Like wind or something.
I think, Prof, I think he means the P's are a bit sharp.
Sorry,
I don't know if
could you
the
repeat that?
P's.
The P's are a little bit sharp. What if I
Yeah,
move that away? Is that better?
yeah, that's much better.
Okay, so I had the microphone too close to my mouth, I think. Okay, so what we have here, if you have a look in the protein that comes off, you Notepad++ to view the protein that comes off, you'll see that these, for instance, these atoms in 5, I think this is from 5-REC, you'll see that in the valine, you can see the first gamma carbon is duplicated in an A and a B conformation, and the second gamma carbon is also duplicated in an A and B conformation. So these duplicate atoms are a problem, and so we need to split it into 2 different conformations, and we can use one of those for the docking process. Okay, certainly, if you use rigid docking, and these were close to the active site, you might run into a problem. But of course, if you do flexible docking, where you allow the protein to be flexible, it doesn't matter which of these conformations you choose. Okay. Right, and then finally, the final thing we did was with the PDB. I said that you could add all hydrogens and merge nonpolar. I like doing it that way in case there are hydrogens to start off with. So if you have hydrogens to start off with, you just want to make sure that all the missing ones are sorted, and so you've got all hydrogens present, and then you merge nonpolar, so you remove all hydrogens that are non-hydrogen bonding. Alternatively, if you have no hydrogens to start off with, you can just add polar only at that point. And then 2 more points is that you need to add Gaskell charges to all atoms, and there's partial charges to all atoms, and then you assign autodoc 4 types. I don't think I've got it here. So you go edit atoms, assign 84 types. Sorry, it is there. And you then save it as a PDBQT. Right, PDBQT. Okay. And then you have a PDBQT that is good for the docking process. Okay. Now, just a little note that this script here, I'm pretty certain that we could get it to run on your laptops. And I'm pretty certain that that script is already there installed when you installed Autodoc tools. It may or may not be. We can check. But it is possible to install it on the laptop. But there's a little work involved in doing so. I find it easier. Everything's available on the cluster, so you can use it on the cluster. And of course, on the cluster, all of this process that we have done on the cluster, you can do it by simply typing. So I don't often, in my course of my research, I don't often open Autodoc tools to do the process, although it's lovely to have Autodoc tools to view what's going on. If I've got a lot of systems, I simply, that whole process of adding nonpolar hydrogens and sorting charges and saving, assigning Autodoc 4 types and saving to PDQT, you can accomplish that using this prepare receptor 4.py script. And so this is a typical command that I would type to do all of that, and I would have a receptor PDQ right at the end. So you can see I type prepare receptor 4, and -R says that the next is going to be the PDB, that the receptor I'm going to use. And so it will create 5rec.pdbqt. And then here's the documentation. And you see with the capital A, the types of repairs I've done. So I've done hydrogens to add hydrogens. And then the cleanup type, the cleanup type I've got. So waters to remove water residues, _. Delete Alt B is to delete the alternate conformations. LPS is to merge charges and to remove lone pairs, and NPHS is to merge charges and remove nonpolar hydrogens. And then -E true says delete every non-standard residue, so it's deleting DMS will go away, so DMS will be deleted automatically. So because I've got this command in my mind, this is what I will type, and I don't have to, if I'm on the cluster, I don't have to copy things. I've got the protein right there, and I can prepare it. But I don't expect you to do that. I think it's quite instructive for you to do everything in Autodoc tools. Yes, there's a question.
Okay, and then Prof, if you were using the terminal like this, how
Yes.
then would you determine the position of your active site? Because
Okay.
how we did it, yeah.
Okay, so what's really interesting is that from Notepad++, or from in a terminal, you can view a file. There's a program called less. If you type less and use space in the file, it actually shows you the file, and you can page up and page down through the file. I can maybe do that when we in our practice session, and then I would then be able to find in the original file, I'd be able to find the ligand. It would have its code, and I would then be able to see the XYZ coordinates of that ligand in that file. Okay. So there's also files on the terminal for finding. There's a grep command, and so grep I can find the ligand that's in there. So I could grep for the code of that particular residue. Okay. Okay. So let's see in the practice session. I'll kind of show you, but please don't feel you have to use the terminal for everything. If you're more comfortable using Autodoc tools, please do it. But of course, if you would really like to try the terminal, we can also try that as well. Okay. Okay. So that there is protein preparation. Okay, I'm just going to stop the share for a moment because I want to get the next slide on the go, and now I'm going to reshare again. Okay. Okay, and so what we've done is we have prepared a ligand. We've got the torsions. It saves as PDQ format. We've prepared the protein. It's got correct charges, atom types, hydrogens, etc. And so we are now ready for the molecular docking procedure. So we're just going to spend a bit of time looking at the algorithms and software in terms of molecular docking. Okay. So please, there are two parts to molecular docking. One, there's a search algorithm, and this really is about searching conformations and positions of the ligand within the search area. But of course, at the on the other hand, you need to be able to score it, and so the scoring function then ranks and says that this is a good docking or this is a bad docking. And yesterday we did 100. I asked Autodoc to do 100 separate tries, and of course it would have had its own scoring function. And of course, the best results are the ones that we are going to actually work with. Okay, so the search algorithm can be systematic. We're going to be using a genetic algorithm, but there's a lot of different ways that it can search molecular dynamics. It can raise the temperature of a ligand, give it energy to change conformation within the active site. But in the end, you have to score it, and you can then at the end of it, you will assess the molecular docking. Okay. There are several different docking programs. If you're fortunate enough to be working with Discovery Studio Suite, there's Ligandfit. Okay. Of course, we all have access to through the CHPC, we have access to the national license for Schrodinger software. So all of us have access to Glide. And were we working in person, I would have been teaching you Glide. So in previous years, I've taken people through Schrodinger to work with Glide. But of course, I don't have access to my computer lab at the moment, so a positive aspect of what we're doing with Autodoc and Autodoc Viner is that these programs are freely available. There's, I think they're free or licensed at no cost, so you will always have access to Autodoc or Autodoc Viner. And please, Autodoc and Autodoc Viner are quite old if you look in terms of time. So there are more recent versions of Autodoc coming through, which I'm not going to use, but I want to make you aware that yes, the principles come through, but don't be tied by this. Always explore the latest in terms of the literature and the software that's out there. Okay. Of course, if you're using Ligandfit, what Ligandfit does is it first has to, you start off, it first, you have to look or define a binding site. And then, of course, it generates a ligand conformation. Does the ligand conformation match the site shape? If it does, then you can dock and optimize it, and you can improve on it and go around. And then you're going to then try again, try a different one. And of course, you do this procedure many, many times for reproducibility. And one of them, the real conformation, if I could call it that, will come up again and again for reproducibility. So the more you do it, the more likely you are to get the correct docking orientation. So their scoring function in Ligandfit is lig score, and the lig score it's an empirical scoring function, and it's come from experiment. So they've defined that lig score as a result of some protein ligand complexes with known experimental KIs have informed it. And so if you use Ligandfit, the scoring function is derived from experimental evidence. Okay. But of course, we're going to use Autodoc tool, and the Autodoc 4 for its post prediction uses a Lamarckian genetic algorithm. And so what they've done is they've mapped conformation into genotypes, and of course then you can from your genotype you can then get a conformation, a real thing in 3D space, and then does it fit very well? If it doesn't, you can change it. You can mutate a genotype to make an actual, I won't go into how this mapping does, but it is a genetic algorithm. They've got a way of encoding. They've got a way of encoding the conformation. So this is not biology. This is 3D space. They've got a way to encode the conformation of your ligand. And of course, they've got a way to decode it. And of course, then we know, will it fit within the active site? And so they can then improve. And of course, it can learn. And that's where the Lamarckian comes. It comes at the learning genetic improvement of that. Of course, with Autodoc 4, there's how do they calculate energies and interactions. But of course, there is a thermodynamic cycle that you have to take into account. What you've got is your ligand, you've got your protein that is unbound, and you need to know what the energy change is between changing your conformation of your ligand to the bound conformation. You need to know the energy cost of changing your protein to the protein's bound conformation, particularly if you're doing flexible docking. Of course, this changes naught if it's rigid, okay? And then, of course, what is the cost of then placing that correct conformation of ligand within the correct conformation of the protein? And then how does it calculate these energy interactions at each stage? Well, this is the energy function. and it should remind you a lot of molecular mechanics. Your free energy cycle is your, you look at your bound and your unbound energies, and of course your change of entropy of conformation. But of course, how those calculated, they're calculated in a way that reminds one very much of molecular mechanics. Other than these empirical numbers, these empirical parameters that you've got here, have a look at the terms. And for your van der Waals, can you see the Lennard-Jones coming through, the 612 coming through? The hydrogen bonding term is something very similar to the Lennard-Jones, but of course you've got your electrostatic term, again coming through from molecular mechanics and calculating energies. Okay, and then of course it has a solvation term, desolvation term that calculates the energy. But how really does Autodoc, how do we work with Autodoc tools? Here's a flow diagram to just give you the flow of how we've worked. Some of this we've done. We've separated the crystal structure into ligand protein. We've done that already. We removed crystal structures, waters. We've done that. We've added we've added polar hydrogens. We've assigned charges in Autodoc times. We've saved the protein as a PDQ2 file. That part is how we dealt with the protein. Then we sorted out the ligand. So the ligand we sorted out as PDQ2. And of course the ligand we got from DFT calculations or from XTB. Okay, right. Then of course we've set the grid center. You have to run AutoGrid 4 to produce grid maps. And then we set the ligand and molecule for docking, and then we run Autodoc. So we're going to run through this. We've been through this entire procedure, but we're going to run through it all again in the practice session. Okay, please just be aware with Autodoc, see these parameters here, these empirical parameters here. Here is a file from Autodoc, and I can barely read it here, but they're the actual values that are used in that equation. And these are individual atoms per, individual parameters per atom that are used. The reason I'm alerting you to this is that this file can be added to. So if you've got an unusual organometallic system that you want to work with, and there's a metal that no one's ever dealt with before, is not present in this, of course, you can intelligently look up literature and get appropriate parameters that will work for your system in here, and you can adjust that file to make it work for your system. That's one thing that I like about Autodoc, is that particularly when you're working with different, so when you're working with different atoms, unusual atoms, Autodoc 4 is my tool of choice because we can always add additional atoms to it. Okay. Of course, another program that I might show you if we get time later on in the week is Autodoc Vina. Please, okay, just hang on. If you got a question, please unmute yourself if you got a question, or raise a hand and I'll listen to it. So with Autodoc Vina, just note their scoring function was based on the PD bind data sets. So the scoring function from Autodoc Vina also is coming from experimental evidence. So it's and so we'll speak a bit about X-score, but of course Vina scores quite nicely. Okay, if you want to look at how the scoring function is set up in Vina, here's some extra reading for you. Okay. Autodoc Vina, I just want you to be aware with Autodoc Vina, what we do is we're going to use exactly the same preparation that we used for Autodoc, and you use exactly the same preparation for both the protein and for the ligand. For the protein, what we've got to do is we've got to get a PDQ-T, and you can use Prepare Receptor 4, or you can use Autodoc MGL tools to do that. And same with the ligand, you can do the same. We've done optimizing the ligand using Gaussian XTB, and we've created PDQ-T of the ligand and the protein. The difference with Autodoc Vina is that you need to have a Notepad++ available, because you're going to have to type a set of commands inside a file, and then we'll be able to run after that. So you actually have to use Notepad++ to set up Viner. And once you've set up Viner, then running it works really nicely. Okay. The type of file that you set up for Autodoc Viner looks like this, and I'll show you, I'll explain it, and we'll try and do a couple of dockings using Viner in this way. It's a little bit more work, I think. You have to make sure in Viner, you have to make sure that you've typed all these names in exactly correctly. If you mistype something, then of course the run fails. It's a bit more work, but the work pays off, because Viner on the cluster, we can set CPU equals 8 on the cluster, and so you can use the power of cluster in docking really to a great extent. So Viner is really nice. Okay, and of course, some of you may be interested later in your projects in using Glide. If you have access to Schrodinger Maestro, in Maestro, you'll see that the procedure for docking in Maestro is not that different from when you work with Autodoc or Vina. You've got to sort out the ligand. And of course, with the ligand, instead of saving as PWQT, if you're using Glide, you use their ligprep tool that does much the same. And of course, you've got to sort out the protein as well. The nice thing about Glide, that I like about Glide, is that you have a crystal structure protein, you go through the protein preparation wizard, but where's your active site for docking? Well, with Glide, you can choose the X-ray ligand, the co-crystallized ligand, to show you where to dock. But what if there's no co-crystallized ligand? Well, it's got the sitemap tool that will search your protein for pockets and say this is likely an active site, and then you know where to dock. And then, of course, afterwards, similarly, like with AutoGrid 4, you've got the grid generation, and then you can do the docking in Glide. Okay, so Glide is another program that can be used for the molecular docking. Okay, we do have a national license for it, so through your supervisor, you probably would be able to get access to the Schrodinger suite. Okay. The nice thing I like about Schrodinger is that Schrodinger has plenty of nodes within the NIME suite of software. NIME is a free tool, but Schrodinger is not. But if you have got, so often in my research group, if we want to set up docking using Glide, this is the way we'll do it, is we've got a worksheet, workspace that looks exactly like this. And we can drag and create a node. And this is a PDB downloaded from RCSB. So 5REC. If you want to get 5REC, I'll open my NIME workspace and I'll put this node in and I'll edit it and I'll make sure it's 5REC. And I'll run this node and I've got 5REC available. And then, of course, I can get a Schrodinger node and drag it in and make a connection, and then that will prepare it. And so my 5REC is now there, and I can then do my grid generation, and that's already from the protein preparations already for docking. And then what about my ligand? Well, we've used Marvin Sketch. So with Marvin Sketch, I can just put the Marvin Sketch node into my NIME workspace. And so when I double click this, I'll be able to, Marvin Sketch will open, and I'll draw my molecule in, and I'll close it, and my molecule is ready for docking. I might have to convert the molecule into a format that's necessary. Might want to view it in 3D space. That's just to view it. That doesn't really aid. But then what I can do is then I can get it ready, use ligprep to set the molecule up for docking. And now I've got my ligand for docking, and I've got my protein for docking. And so I can then organize this. Of course, in Glide grid generation, at that point, I will tell it X, Y, and Z coordinates for the active site. And so that when the docking occurs, it will go into the active site. And of course, I can have another node that will help me visualize what's going on. But this workspace here is really nice if you've got hundreds of ligands. I've done this before. I've started with hundreds of ligands, and it doesn't change the workspace. The hundreds of ligands go down these pipes, and so at the end, I've got hundreds of dockings at the end of it. So Glide is very powerful, particularly if you want to do high throughput work. If you've got a lot of systems to deal with, Glide within the context of the KNIME workspace, we can do molecular docking. Okay, I'm leaving it there. We're almost out of time, so can we meet in 10 minutes? If you've got questions, ask them in 10 minutes time, and we will, I will be able to answer them in the practice session. Okay.